It's okay. It is. Okay, nos with that. Croeso Constian I gyd I Brifysgol Bangor rwy'n falch iawn I weld nifer o fyfyrwyr, cydweithwyr a cyfeillion yma ar y cluster arbennig hwn. Well, good evening and a very warm welcome to you all to Bangor University, and I'm delighted to see so many students, colleagues and friends on this very important occasion. Tonight we are inaugurating the Professor Dennis Crisp Memorial Lecture Series in Marine Sciences. And the profound influence that Dennis Crisp had on the School of Ocean Sciences cannot be overstated. And this series intends to recognise and to honour his remarkable legacy. Beginning tonight and going forward on an annual basis, we will be inviting back to Bangor some of the PhD students who studied under Dennis and who have now gone on to outstanding academic careers. And I can tell you I have met many across the world. We're asking them to present to today's students and to our colleagues on their research areas, and also to reflect on how Dennis influenced their development as academics. I would like to especially welcome Dennis's family here tonight and to thank them for coming all the way here back to North Wales. His daughter, Mary Wheater, son and daughter-in-law, Graham and Jill Crisp, and his granddaughter, Sarah Crisp. You're all most welcome, and we hope you enjoy the programme. Tonight we will hear from one of Dennis's most accomplished students, Professor Donald Manahan. And the Dean, Colin Jago, will introduce Donald, but I'd like to thank him and his wife, Nicole, very much for making the long journey from Los Angeles to be with us here tonight, and for being the inspiration, indeed, behind the whole idea of this series through his enthusiasm for his former mentor. And I'd now like to invite the Dean, Professor Colin Jago, Dean of the College of Natural Sciences, to introduce Professor Mann.
Dennis Crisp was a major contributor to the development of ocean science at Bangor University, arguably the major contributor. Dennis was not only an exceptional scientist, and that's amply evidenced by his publications and honours, he was also an expert strategist and tactician who developed a coherent vision for ocean sciences at Bangor, and then supplied the energy and the skill to bring that vision to fruition. Under his leadership, the Marine Science Laboratories, as it was then called, developed into one of the leading marine science centres, not just in the UK, but in the world. During his years of leadership, the laboratories grew an international reputation that attracted talented young researchers from all over the world. The laboratories grew physically with new buildings and facilities, and they acquired a new research vessel, the Prince Madog, which was the largest such vessel in the UK university sector. Hence, Bangor, or more accurately, Menai Bridge, became a world centre for marine science. Dennis was an exceptional man. His approach to science was dedicated and uncompromising, and his approach to everyday life was much the same. This had the consequence that he was much respected by his research students and collaborators, to whom he gave unqualified support. But it also gave rise to the consequence that he was not greatly appreciated by officialdom and administrators, who he generally regarded as an interfering nuisance and a waste of time. The sort of adjectives used to describe Dennis ranged from brilliant, inspirational, energetic, to irascible, pugnacious, and downright abusive. which adjective you used depended upon who you were. So fellow scientists and the staff of marine biology erred towards the descriptors at the front end of that list. So inspirational, brilliant. But bureaucrats, and I have to say the central management of this university, tended to use the adjectives at the end of the list. Irascible and pugnacious. But it was Dennis's energy and pugnacity that did contribute so much to the growth of marine science in the university. And Dennis provided the foundation on which the next generation of marine scientists has built. And it's particularly gratifying to see members of his family present tonight to witness the high esteem in which Dennis is held here in the university. One measure of success for a university researcher is in training the next generation of eminent researchers. And by this measure, Dennis Crisp was certainly successful, sprinkling talented young scientists across the world. And tonight's guest speaker is an excellent example of this. Professor Donald Manahan came to Menai Bridge with a first degree in zoology from his native Ireland and worked with Dennis on the environmental physiology of marine invertebrates for his PhD. He then moved to California and sadly never came back. Donald is now a professor of biological sciences at the University of Southern California in Los Angeles. His area of research is the environmental physiology and adaptation of marine animals, specifically marine invertebrates and their developmental biology in different environments. So temperate environments, polar, tropical and deep sea. He's particularly interested in the larval stage of development that is intermediate between the egg and the adult stage of the majority of marine animals. Understanding the growth and development of any animal in its environment is a complex process, but for the larvae of marine animals, which respond on the one hand to small turbulent eddies on centimetre scales, and on the other hand to oceanic currents moving over thousands of kilometres, and to timescales from seconds to years, the complexity is considerably increased. Donald's work has added hugely to our understanding of how larvae work. He has shown how species develop very different life history strategies in different oceanic environments. For example, the cold polar oceans of Antarctica, the warmer temperate oceans of California, and deep sea environments. Such research is crucial to our overall understanding of how marine ecosystems work and how they will respond to global environmental change. Donald has led over 20 scientific expeditions in the Antarctic and in the Pacific Ocean. He served as chair of the US National Academy of Sciences Polar Research Board and on the National Science Foundation Decadal Planning Committee for Ocean Sciences. He's also served on federal advisory committees to the National Science Foundation. And in 2000, the year 2000, a mountain in Antarctica was named Manahan Peak in honour of Donald's contributions to research and education on that continent. In 2001, he was appointed as a lifetime National Associate of the US National Academies in recognition of his services to the nation in matters of science. And in 2011, he was elected as a Fellow of the American Association for the Advancement of Science for his contributions to the study and physiology of animal development. And these achievements demonstrate beyond doubt that he shares with Dennis Crisp an ability to make very significant advances in marine biology. in our understanding of marine organisms from molecular to ecosystem scales. He is, though, much more tactful and diplomatic in his dealings with officialdom than Dennis ever was. We are greatly appreciative that Donald has taken the long trip from California to be with us tonight, and I would now invite him to give tonight's lecture on Dennis Crisp, a visionary for global change.
Thank you. Thank you, Dean Colin Jaco, and of course, Vice Chancellor John Hughes, and many friends and many guests. This has already been an amazingly emotional night for me. I've been meeting people in the reception I haven't seen for 30 plus years. As a professor, I've given probably thousands of lectures, but none, I can say, none mean as much to me as this. For it was at Bangor that everything I have done for the last 30 years was developed. It was at Bangor where the ideas of how to approach complexities in studying the life sciences, the fearlessness of the team that trained me when I was at Bangor, anything was possible. Try it out, give it a go, dream big. Those were the kind of phrases. It wasn't just Professor Crisp, but a whole team of others, many of whom are here today, that helped me think about things on scales that were just unimaginable when I was a young kid at age 22 and got off that ferry from Dublin, Trinity College Dublin, to arrive on Bangor, to a life-changing experience. And that's the most important reason that I flew all the way from Los Angeles, California, to be here today. It's also the 100th anniversary, as the slide shows, of when Professor Crisp was born. 100 years. It doesn't seem possible to me that he would have turned 100 this year. Because when I met him, when I worked with him, even in his 60s and into his 70s, he was like this kid in a candy box, as they often say in America, just excited and positive about so much and so many things. Colin just talked about the different facets of Professor Crisp. I won't comment on those. I will just talk to him as what I consider this genius for seeing the future, and I'll talk about why I think there's evidence to suggest that even 100 years later. This might be the younger Crisp, the more feisty one. Here's the Dennis Crisp, the little lighter side of him. And one thing, this is Thanksgiving Day in America, and I'm here with you instead of being with my own family in America. And Dennis had a love of American science. I met him many times at conferences after I moved to America to work there. And he spent a lot of time in that latter part of his career attending conferences. To this day, his name is really very famous in America. In fact, when the invitation went out, and I want to personally thank Sheila and her team for doing an incredible job of putting this event together, when the email went out from Bangor University to many alumni across America, emails were flying around, phone calls were happening. There was incredible excitement in America. Across, literally, I'm in California, Oregon, Connecticut, Maine, across the country about this event and the celebration of somebody that many today in America know had an enormous influence on the field in general and specifically. So I'm here representing all of those individuals, I feel, in America. His visit to America, one of his last visits was 100 years ago, 1986. And I wanted to bring this to your attention because I think it's very nostalgic. He was well known for making lists, and here you see this is a note he left behind in his hotel room saying, "Remember to bring lunch, glasses, teeth." I'm sure his son, daughter will remember those days. And then a little joke showing the kindness of the man. His wife, Ella, put a little handwritten note on that list saying, "Kiss wife." That note is with me today. For 30 years, it stayed in a storage box in Sandy Shumway. Some of you remember Sandy, another student, stayed in her house. And as we were talking about this, she works in Connecticut, I work in California. She said, I know, that must be somewhere. And she looked and looked and looked and looked and looked and looked. And then she wrote this email, bingo, I found it. And I want to give it to the Crisp family because it has your father and your mother noted on this. And it has come all the way from America this evening. You're very welcome. And there's a personal note to you in there, Mary, from Sandy Shumway. So went from Connecticut to California, from California to here. It was left behind, and not knowing its significance at the time, Sandy kept it. What a sweet note. The famous list maker, and then his wife putting in, don't forget to kiss me. This was the last time I met Professor Crisp in 1988 at a Gordon conference. Dennis can be seen here, the only one with a tie. Those of you who are microbiologists might recognize this famous microbiologist, Farouk Azam. Pete Gabbett, you'll recognize or know this name. This is Peter Hachachka. So there were a lot of real big-time middle-career players at this conference approximately 30 years ago. And then there was this young kid here in the center. That's me with the darker hair. The sun was in my eyes, so I turned it down. But I remember how proud I was. I'd been in America about 8 years by then. I remember how proud I was that Crisp was nailing the damn thing at every single talk. This is the Gordon Conference. This is one of the most distinguished conferences that thinks about chemical biology in the United States. Huge endowment. Can fly in anybody from anywhere. And they flew in Dennis for this. And their questions would go on. He'd be bam, bam, seeing a hole in an argument, seeing it a different way. I was so proud to be his student, even though at that time I was his former student. And to see this sort of the 40-something-year-old crowd, I was just in my early 30s back then, or even earlier, the point, to see that how well regarded he was in America, and to see how sharp he was, even at age 72. And sadly, he died just 2 years later, so it was the last trip to America. His legacy goes on, and that's the purpose of this lecture series. And so I wanted to give this a bit of a sense of how powerful this legacy is. This is one of his most famous scientific papers, published in 1962. As Dennis told me himself, and I'm sure he mentioned to others in the room, that he felt this was the paper that got him elected as a Fellow of the Royal Society. It was this cross-disciplinary thinking between chemistry and biology, which was revolutionary at the time, that put him on the map, and then helped put Bangor on the map in terms of thinking about this interdisciplinary approach to thinking about science. Bramble, the famous Bramble, after whom buildings are named here, was, of course, a mentor to Crisp. Bramble actually was Irish-born and came, I believe, did his undergraduate degree in Trinity. In fact, I went to check on that 2 days ago when I was in Dublin, stopped by to see a former student of Bramble who's just retiring. So Bramble's name is very active, and Bramble had a big role in bringing Crisp in and supporting these kinds of intellectual initiatives all those years ago. But to be more quantitative about it, I looked up all of Crisp's papers and got the sort of digital librarians to help me put this little segment together. These are all the papers that came from that 1962 publication. There's hundreds and hundreds of them. It started an entire field. One paper started an entire field thinking about the chemical basis of chemoreception and how it would induce change and metamorphosis in animals. In fact, one of these papers, if Dennis was here today, he would love. It was just published a few weeks ago in the National Proceedings of the National Academy of Sciences, one of the most prestigious journals in the United States of America. And the part I've underlined in red here says, this one molecule that Crisp had helped work on, and others in this room had helped work on too, has had this whole new life, as illustrated in this paper in 2016. Because predators now key in on this molecule, knowing that eventually some goodies are going to come in, because that's where they will undergo metamorphosis. And so the concept of what people are calling keystone molecules in setting entire ecosystems, a really huge idea. National Academy of Sciences. One molecule punches in with effects that are far greater than originally intended, is the sentence that caught my attention, by my colleague, Professor Richard Zimmer of the University of California. Quantitatively, you can look at things in really sophisticated ways. The chief librarians helped me put this together. We say, oh, he published a lot, he did this, but what did it mean? Did anybody care? So here's how you analyze care if you're a librarian. It's called historiograms. And you look to see how one paper influenced another and influenced another. It's called the web of science. And here at the top is the Crisp and Meadow paper of 1962. And you can add more papers. Here's like the top 100 papers that are cited in the citation index. And you see the web of science gets even more complicated. And here's 200 papers, and it gets even more complicated. And this is how modern information moves around. It's not just one person looking at a book in a library anymore. It's highly quantitative digital interactions with very sophisticated software. To even do the run on this took a big supercomputer several minutes to do. So this is the modern sense. Crisp would love this, I think. He'd love to see the influence continuing decades and decades after he did the original work. And you can start to peel back layers, as shown here, and start to see a new field starting to emerge here. What is this field about? Well, this field was Crisp thinking about climate change in the 1960s. It was the effect of a very severe winter of 62 and 63 on marine life in Britain. Here is the paper. And then all this idea started to emerge out of this. He used whales soon after he arrived here as a perfect natural laboratory. Here's all his sample sites. A perfect natural laboratory to be able to understand how quickly life can collapse under environmental change, weather change. And he wrote a very important paper, which is very, very, very highly cited. And one of his co-authors is in the audience this evening, Craig Kensler. Craig, would you stand up and just acknowledge that you're there? You go. Hi, Craig. Craig Kensler, in 1962, I believe, when he came to Bangor, was Crisp's first American, 61, was Crisp's first American student. And Craig Kensler hired me as an intern undergraduate decades later in Ireland, and that's how I made my connections with Bangor. So one generation, the next generation. An incredible paper. I read your paper, Craig. You ought to go back and take a look at it again. In the modern light of climate change, it really says a lot. Because what Craig and Dennis and others were thinking about, and here's a brilliant paper by Dennis Crisp. Note the date, 1959. And the title is The Influence of Climate Changes on Animals and Plants. Ha! Anybody heard of climate change? Anybody concerned about it? He was thinking about these things in the 1950s. And the line I've underlined here is that winter temperatures have risen. He did this huge analysis of the statistical data for this part of the world and elsewhere, and started to see trends in the 1950s for shorter, warmer winters. 1959. So my title today is, he is a visionary for global environmental change. Even though you think of him, as Professor David Thomas and I were just talking about, as thinking about barnacles and metamorphosis and settlement, he had this other whole side of his publications, thinking very broadly, soon after he came to Bangor. Colin, I liked your introduction about him, and here is another way to think about it. This is on page 26 of a book that was written called Across the Bridge. It said, "For better or worse, things would never be the same." When Dennis Crisp was hired, July 1951. Now, imagine, it's only a few years after the end of the Second World War. And this is to students in the audience. As a dean of students at a big university where I work, there's a lot of pessimism at times. Oh, it's hard, it's this. Just so, look what Crisp did. A few years after the war, his first job. They promised him a big building, and they said, well, we can't afford it right now. We have instead some money to buy this little private house on the Menai Bridge, which is Westbury Mount. Many of us held our offices in there. Instead of going, oh, boo, you promised me the world and you're giving me this rundown little house that isn't even designed to be a research facility. He got to work and made it a research facility. Not only did he make it a research facility, he made it within 10 years one of the best in the world. In the world. That drew Craig Kensler from California to come to work with him. That drew me decades later from Ireland and hundreds of others from around the world. Damn. How did he do that? Damn, how did he do that? Read about it. There's a wonderful write-up about it. And the students, I'd highly recommend read this book. I read the whole thing from cover to cover again for tonight's lecture. It was published some years ago, but with a different view. It's called Across the Bridge. Because what Chris undertook was something really monumental. To build a research team, as the first director, to take on problems as big as this. We can never, as a society, walk away from the study of the oceans, simply because of this. It's the biggest environment on the planet. Now, you all know it's 3/4 of the surface of the planet. Perhaps you don't know by volume, it's 99.5% of where anything can live on the planet is in the world's oceans, 99.5. In fact, you could forget terrestrial biology. Oops, we live there. So maybe not, right? But the point is the scale of the problems, and it's beautifully put by Arthur Clarke, the great author and futurist, in this quote that I've taken from him, how inappropriate to call this planet Earth. It should be named Planet Ocean. Somehow, Crisp, even though his background was really not in marine science, it was in chemistry and mathematics from Cambridge University, got that. Today we know, as shown at the bottom of this, is that the oceans regulate our climate and our weather. They provide about half of the oxygen we breathe. So just think about that. Take first breath, take the second breath. The second one is all from the ocean, or 50-50, depending where the molecules actually come from. Half of the oxygen that sustains this planet and us comes from the oceans. Mess with that, there'll be dire consequences. And not only that, it provides us with food and energy and minerals. So oceans are a very, very big thing, but they're really hard to study. You can't just walk out of the field and take a sample like you can on land. They're very hard to study. And when all this was built, this spoiled kid from Dublin, me, showed up 30 years later, right? Walked into this unbelievable facility where, as I said, anything was possible. This paper that I published with Dennis is one of my proudest papers of my career. It was published just after I finished my doctorate. It's a simple, dull, boring title. That doesn't really matter. What really matters was inside this were all of these sophisticated tools in radiochemistry, biochemistry, physiology, culturing, all of these tools that have basically laid down the foundation. It's exactly what a great PhD should do, lay down the foundation that no matter how things changed, you were able to adapt and have the toolkit to address it. And so hence, to Bangor, I say, I love you. This is actually on the front page of the Bangor magazine. Not a phrase scientists use very often, but it came up, and there you are. I mean, to me, my start in Bangor led me around the world, to the ends of the world, literally to the South Pole, to the bottom of the world, the bottom deepest oceans that are miles deep, and I'll tell you a little bit more about that in a moment. But I do want to say that what was envisaged here was world-class. This concept, this NERC unit that Pete Gabbett was in, and Graham, and others who were, and Andy Beaumont, others who are in the room here this evening. This unit was sort of so sophisticated. I'll call it like the Max Planck equivalent of a marine institute in its thinking. And that is, it put together experts, brought in people who didn't have backgrounds necessarily in the study of the ocean. They came in from plant biochemistry, as Pete Gabbett did, and others. And they sort of put together this world-class team to address complicated issues that spanned from whole organisms all the way down to subcellular levels of analysis, simultaneously. I mean, marine biochemistry, in my opinion, was almost invented here in Bangor. And yet today it's a standard tool used all over the world. In fact, interestingly, all of my service, I suppose I will call it, to the National Science Foundation over the last 20 years. If you look at what NSF, and which is a huge operation in the United States of America, is still trying to do the same kind of thing. It's trying to take an interdisciplinary approach from studying physics, chemistry, biology, mathematics, and then looking at biology at all the levels of analysis from a whole organism down to individual molecules. It's called modern integrative science, but in my opinion, there was no better unit in the world than was formed here in Bangor. Why do I say that with some confidence is the key problem when you're studying the biggest environment on the planet is most of the stuff you can't really study. You can count it, but you can't study it in any mechanistic way. You can't culture things. And it was Dennis's ability and understanding of the importance of culturing things that really formed the basis of what this famous place was about. If you can't culture things, it's very hard to study them. Many Nobel Prizes were given because people were being able to culture things. One studying culturing sea urchins, or culturing fruit flies, or culturing nematode worms, just to use a few examples. If you can culture them, you can start to do highly and deep understanding. And these are some of the important breakthroughs that started to happen in marine science. In some ways, Crisp invented what I will call the fruit fly of the ocean, this concept that you could start to do things in a mechanistic way that had, of course, had a whole history down to the famous Thomas Hunt Morgan, for those of you who are geneticists, who pioneered this kind of thinking in the 1920s and 30s when he was at Caltech. But it's very, very, very hard to do. Here's a little short video. Let me turn the lights down. Come on, come on, come on. It's not coming back up for me. Oh, there we go. Thank you. What you're seeing here are an embryo, about the same size as you all started, by the way, about the size of a human hair, which is illustrated here. But they're very hard to study because they have behavior, they move around, they sit, they swim. So it's not like studying microbial processes where they're all a unified population. It's a very diverse population that gives you high error. And Crisp and his team really sort of helped to figure out how to approach those kinds of questions. So what I'd like to do for my remaining time we do this evening is to honor Professor Crisp, as was in the beautifully well-written, in my opinion, email invitation for tonight's lecture, was that the diaspora of Dennis, including myself and hundreds of others, have been asked to return to Bangor and give you some ideas of where science is today and how that was all founded here at Bangor by Crisp and others at Menai Bridge. So I thought, I've done a few things in the last 30 years, and I just wanted to give you one or two examples. But the examples I'm choosing, I'm pretty sure Dennis would have liked. One, I know he would have liked, because we talked about it just before his death. So I picked my most difficult project of my entire career to talk about. It took about 8 years to do, and I'll talk about it, and if I get the timing right, in 5 minutes or less. Here's the challenge. The challenge is, yes, the oceans are huge, but most of the stuff is down here. It's very deep, and it's very cold, and almost nobody goes there. It's called the deep, deep ocean. And around the time that Crisp was, at that point in his 60s, a huge discovery was made in ocean sciences. Today we call it the hydrothermal vents. And it was a fusion of geology and geophysics and geochemistry and biology and deep time evolution, a really massive new sets of ideas. These vents were discovered miles down. Hot chemical-rich water was spewing out of the center of planet Earth. People were calling it the primordial soup dispenser. And it started to balance things on the planet that people hadn't thought of before. A lot of the iron that fertilizes the oceans, that gives us this oxygen and the plant life, comes from those regulations of those deep hydrothermal vent systems. Crisp was fascinated by this stuff. And that's why I know he would like this brief study. Because remember, he told me, darn, I wonder how the larvae work down there. Do you think you could ever culture them? I said, Dennis, professor, actually, I used to call him. I only called him Dennis when he came to America. He was always Prof. So I'll start calling him Prof this evening, in case he was here. So Prof, no, it's 250 atmospheres down under pressure. There's no way you're going to figure out how to culture these things. It's really, really hard. Here's the site, shown in red here, where we went ahead and did this. Now, this site's very important. What you're seeing here is tectonic plate drift, speeded up millions and millions of times. And what you're seeing, which is particularly interesting, is you're having a look here at, step out so I can point it out. You're starting to see the mid-Atlantic ridge form, Antarctica starting to form, the other Pacific and American plates are starting to form. Watch India. This one always interests me. Look at this thing come out like a bat out of hell. Boom, 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 boom. And hits so hard it makes the Himalayas. A lot of connections with the Himalayas and Bangor, I remember. Former principals here. And boof. And here's what it looks like today. Now what matters is that at these tectonic spreading plates, now remember tectonic plates as a theory was really not universally accepted until quite late into the 20th century. You'd agree with that statement, Colin. Yeah. So the discovery of hydrothermal vents was huge in science for the definitive, unquestionable evidence that these plates existed. And the excitement among science was the following. Wow, maybe this is where life started. Maybe this primordial ooze that's coming out of the center of the earth, perhaps that's the history of life. And people are going, yeah, but we've never found it. So there's a lot of skepticism about those ideas. But they were right. Paper published in Nature last year. Look where they had to go to do the study. Way up here, north of Norway, between Iceland. And there they found a set of organisms at depths of 3,000 meters, the blue stuff, the dark, deep geophysical mapping. They found these microbes, and when you sequence all of their genes, it's the best evidence on the planet of the transition from microbial life on the planet to what we are, complex multicellular eukaryotes. Ah, was this an incredible paper in Nature. Found at these extreme events, found at the primordial ooze, so to speak, of what we think evidence is for where life was formed. But it's really hard to get there. You're entering the deep ocean abyss, miles, miles down. If you want to, you can do silly things like this. In the 1930s, you kind of lock yourself into a sphere and they hold you over the side. Hopefully the little string doesn't break and you go, that's as far as we were able to do anything in terms of understanding these deep environments. Today, it's a little more sophisticated. It's still a ball, and I spent many, many, many hours inside this ball. It's the titanium sphere that goes inside the submarine Alvin. Look, here's it's being made. Incredible engineering to make this thing to handle pressures of 300 atmospheres pressure. And make sure it doesn't leak or goodbye. And a typical day when you're working on these things, this is me here. I was the chief scientist on one of these cruises. It's amazing. Everybody's up and excited like hell. It's 8 o'clock, 6 o'clock in the morning. The team has been working on it all night. Out you go. You stuff you in here. It doesn't look like you're being stuffed that much, but there you are. You're being shoved into this tiny little sphere with computers and equipment, and it's like, good luck. Maybe we'll see you tomorrow. Off it goes. Of course, the dive I was on was number 3,132. I still remember that number to this day. So it's been very successful for decades and decades and decades. And what it was after was this, mapping what Dennis was interested in. Here is one ridge, and then, of course, there's an earthquake, if I may use that phrase, and there's a shift. And the question is that if life is here, how does it get up to here? I mean, how do you travel from Bangor to London, or Paris, or for that matter, from London to Tokyo? How do the propagules cover these kinds of distances? It was one of the great, great mysteries. Dennis spotted it right away. It was beyond his possibility to study it, but the tools and techniques I learned at Bangor, boom, we cracked the question in what is now, to this day, my most cited paper in Nature, is this simple paper called Larval Dispersal Potential. And the stuff that I underline here in red are things that really I got to think about all those years before in Bangor. It was studying physiology and development and thinking hard about hydrodynamic information. I remember John Simpson being kind of the god of that when I was there at Menai Bridge. I wasn't good enough to be able to think about the physics of these things, but years later, working with scientists at Woods Hole, where we collected literally millions of data points over years to look at the current movement in these deep sea environments, and then we could make mathematical models very crispy, and make mathematical models of whether, if the larvae could last this long or this long, where would it go, could it disperse, and the question was cracked in that particular Nature paper. Students, the questions are still wide open. Nobody's published anything since then, but it does work. You can culture things at super high pressures and low temperatures. Here's how you do it. Become a chief scientist, have 2 ships at your disposal with a few hundred scientists, be given a submarine. That was the coolest thing. Talk about, toys. Given a submarine, you decide where it goes, what it does. This is by the way, the same submarine that found the Titanic, Alvin. Amazing machine. And as I said, you're scrunched into this little ball at the front. This diagram here illustrates the fact that here's one hydrothermal vent and here's another. I mean, obviously it's a diagrammatic one, and the concept of this collapsed caldera here is how do organisms disperse up and down these issues, because they change very quickly. As soon as there's tiny venting geological changes, little populations get wiped out, and how do they reform very quickly? When you come into these things, it's pretty amazing, because the temperature of the water that's coming out at the center of the Earth is about 400 centigrade. So you're going in this submarine, and the pilot, not me, the pilot better be really good, because you're going in inch by inch. You have temperature probes in front of you. If you get in too far, everybody gets fried. If you don't get in far enough, and the organisms are right on the edge. They're living on the edge of just the right temperature, and not too hot, not too cold, or they don't get all the geochemical energy that, of course, is the basis of these environments. Culturing these things was a blast. And I say that in the most challenging way. It took us 4 years to work out how to culture. Pete Gabbett and I were just talking about this over at the reception a moment ago. Pete, you had to culture little tiny things at 250 atmospheres, measure respiration rates of little tiny things under 250 atmospheres, and blah, 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 blah. So it was enormously difficult to do. And in fact, when we tried to propose to do it, I remember all this huge negative reviewer mass saying impossible to culture. And so we made a little homemade video here, which I put on my website, because I don't know who the reviewers are, but they know who they are. And I said, yes, it can be done. Let me just show you. So what you're seeing here, it's not the best video. It's just done by me. But this is the first time ever that these organisms had been cultured from these deep sea vents. They were brought to the surface. What's particularly important is that they were cultured from fertilization to the ciliated larvae for the first time ever. Students, if you're interested in this, there's a little more information about it on my website. Cultured at 250 atmosphere. And interestingly, against all predictions, they only last about a month. And as soon as we looked at the physical modeling, that was exactly the amount of time that would be needed to not get swept off the ridge vent systems. And it turns out the following: this 38-day calculation was predicted right out of CRISP and Holland and Gabbett, biochemical and respiration energetics, predicting the lifespan, putting it into these very complicated physical flow models, and poof, guess what? The butterfly can fly, the bumblebee can fly. But they didn't know how it was done until we were able to do that particular study. Turn the lights back up if I may, thank you. So briefly, I want to talk about my most surprising study that I think Dennis would have liked. The one was difficult. This was not as difficult, but it was really surprising. It's in Antarctica, a place that very few people get to study and work in. It is, in fact, a miserable place to work. It holds the world record for the coldest place on Earth. Little typo here on the Guardian article about it, but if you do the calculations, it's minus 94. That's the coldest place on Earth. It's hard to work at minus 94. I've never worked at minus 94, but I've worked at like minus 60, minus 70 occasionally. It's a tough place to work. But my goodness, is it eerie. When you get off those big monster planes, the pilots can land these things on ice sheets, as you're seeing here. It's like this big dragon, Welsh dragon, spewing heat and fire and flames. Absolutely amazing, the skill set of what it takes to do this. To stand at the South Pole was one of the most profound things I've ever done professionally in my life. It was done on January 17th. And the way that date matters so much is not the year, but the date, January 17th. Because on January 17th, many years before, Scott And his party stood exactly where I stood. And I had read all his diaries. I'd read all the diaries of his chief scientists. I knew what they had done to get there. I got there in about 2.5 hours on a comfortable warm plane. It took them 3 months of slogging, each man pulling hundreds of pounds, climbing to get there. And I think this is one of the most poignant photographs ever of science in Antarctica. It's Scott and his party at the South Pole, just realizing that they'd been beaten by a month or so by the Norwegians. This famous man here is Welsh-born Petty Officer Evans. Look how strong he looked, even there. Scott loved him. He was so strong. Petty Officer Evans. He was so strong, he needed more food than everybody. So the tragedy is he was the first to die. A month later, he died. A month after that, this man died. This is Captain Oates, March 17th. Walked out of his tent, leaving these 3 men, Scott and Wilson. Wilson was this modest chief scientist. And when you read his diaries of what it was like that day, January 17th, he said, it blew force 4 to 6 all day in our teeth with temperatures of minus 22. The coldest March I ever remember. Now that's saying something from the chief scientist who led The Worst Journey in the World, the famous book. Scott's been much criticized for almost, at least 100 years, that's central, about whining, if I may use Huntford's word in his books, whining about being cold in Antarctica. But Scott was right. It was damn cold. It was colder than it has ever been that particular year. We know that because modern scientists have mapped the temperature, because it's the same flight route that America takes to get to the South Pole. They've mapped it year after year after year for flight safety operations, and this year really, really was cold. So Wilson was right, and he was just judging it based upon how miserably cold it was that day. Another great scientist at Bangor who had a big influence on me was Tony Fogg, fellow of the Royal Society, lecturing here in this picture at Bangor University. He wrote this brilliant book, and students, I highly recommend you read it. It's called A History of Antarctic Science. Not explorers, but the science. And I've taken this book with me many times to Antarctica to think about the early scientists who were there before me, who started to think about issues about climate change that are now front page news to this day. It's a lot easier to get there today. This is the airplane that flew us there, along with somebody you will know well, Sir David, with a lot of Bangor connections. He was 84 on this flight with me about 5 years ago. He just turned 90, as you know. Isn't that marvelous pictures of him with the Queen laughing and giggling over a cup of tea the other day? We were so blown away with his energy and drive and enthusiasm. If you look behind, there's this guy fast asleep right here. Not Sir David. He was like, and this was called a boomerang flight. We flew 3,000 miles to Antarctica. The pilot said, nah, we're not going to take the risk of landing on the ice. Let's go back. Flew all the way back to New Zealand again. It's called a boomerang. It's the living hell of flights. Not Sir David. He was totally okay with it all. Even your chief scientist, me, was fast asleep in the back of the plane after a couple of these things. So marvelous who you get to meet on these trips. And of course, as you know, as I hope you've seen the Frozen Planet series that emerged out of his trip to Antarctica and many others. Remarkable it was. And how interested he was in the science and meeting with the scientists during that trip. So here's me in a slightly different suit than today, in a scuba suit. But what I wanted to bring to your attention is this collapsing and opening and collapsing. This is the Antarctic ice sheet that the whole world is watching for climate change. This is the annual ice that melts and freezes and melts every year. You can think of it oceanographically like a giant heart. It just pumps once, boof, and it drives deep ocean circulation, and it does it the next year and next year. So think of it as a huge heart, boof, where billions and billions of gallons of cold, salty water are moved. And if that did not happen, here in Northern Europe, we'd be frozen to death. It's happened before. When these pumps stop north and south, and the oceanography stops. This is all pretty recent stuff that was worked out in the last decade or so, or certainly after CRISP had any ideas about these things. It does suggest something else I want you to remember. Students, this will be on the exam later tonight, that in fact this is a cold atmosphere. This is a cold planet. This is not a warm place. We like it to be warm, but life on Earth on planet lives pretty much at pretty cold temperatures. About 90% of it is ice water. So that's what drew me there. Was I had an education just thinking mostly about stuff that was pretty warm, but realizing everything I'd been told about biochemistry, put a test tube on ice, the reaction stopped. Well, how will that work in the cold biosphere? So that was what drew me there in the first place. The surprise was how difficult it was for us to culture things. I'm holding these little baby embryos in my hand here. But the real surprise was this paper we published in Science that got quite a bit of attention at the time, because it was a totally different kind of biochemistry metabolically. Life down there was driven by just two processes. Now, you all think about this as you're hoping I'll shut up soon and go to dinner, is that the whole process of how you fuel metabolism and what you eat and how you make your proteins is all encompassed here. What's very interesting is that most of it, anybody who worries about salt in their diet, these organisms spend a huge amount of energy, like 80% of their energy dealing with salt. And in most organisms around the planet, it's more like 10 or 20 or 30%. In a human, that's up to 30. And protein synthesis is a tiny part of metabolism. But yet, the real surprise, and that's why I use the phrase that I think Dennis would have loved, is that the rate processes in the cold at -2 are so temperature compensated, it's like being in the tropics. The turnover rate of these proteins is so fast in the cold that it violated what we thought at the time were simple laws of energetics and thermodynamics. As we looked into all of that, certainly more than you want to hear tonight, they don't violate any laws. They just have clever ways of doing it. They turn their RNA, their DNA, they take genes and turn them into proteins at super fast speeds, and yet they grow very slowly. This has a lot of important implications for ideas that are now emerging about life on Earth, because everything we know about what we call the Cambrian explosion in geology happened soon after the end of the last snowball Earth event. Soon being 10, 20, 50 million years, but on the time scales of billions of years of life on Earth, this was very recent. So it had a lot of interest trying to understand, if you're going to evolve quickly, you've got to be able to turn over proteins and DNA quickly. And so cold organisms seem to have that capability. I was really struck a few weeks ago with the recent Nobel Prize in Physiology and Medicine, which in another way was addressing this very question. It is a Nobel Prize for studies on protein metabolism. And out of this study has emerged something that you all think about every day, dinner. It's like, you don't eat enough protein. You certainly don't. Nobody eats enough protein. Nobody. You only eat about, say 70 grams per day, and you need 300 to live. How the hell does that work? Nobel Prize. 6 weeks ago. Incredible work. The concept is called protein turnover. It's this sophisticated machinery that you recycle in your own body most of these proteins to make new ones. That's the trick. That's what won the Nobel Prize. And when we went to these extreme environments, we realized that they had the best examples actually in animal science at the time, still at the time, of having this huge ability to recycle proteins for super low costs. I was so excited to read that Nobel Prize. Look, Dennis used to talk a lot about the importance of marine organisms. So in his honor, I read every single Nobel Prize ever awarded and pulled out every single one of them for tonight's talk. These are all the Nobel Prizes for studying marine biology. It starts down here in 1908 for people studying starfish to study the evolution of human immunity, right up to studying jellyfish. Cell biologists purified a protein called green fluorescent protein out of that thing. This green fluorescent protein allows you to study cell signaling. All the Nobel Prizes in marine organisms are here. Students of microbiology, students who study phytoplankton, there isn't one of those groups on here yet. Think hard about that. What are the foundational studies from studying life in extreme environments? We call it extreme, but of course, as I think I've tried to illustrate, it's normal. We're extreme by having made the water-to-land transition. I think Chris would have liked that. So what next? Well, in conclusion, as I said, I consider CRISP a great visionary for a term that we have today, global environmental change. Here's his study in the 1950s. Front page news just a week ago by the best group in the world who thinks about this, the NASA Cryospheric Science Laboratory at the Flight Center, with billions of dollars at their disposal for satellites, measuring temperature. And here's from 1950, when CRISP came to Bangor and started to be concerned about climate change, all the way up to their analysis last year, 2015. Everything you see in red here means it's pretty simple. The analysis is not, but the picture is. It's getting warmer, folks. No matter what any elected president may tell you. Here's why. CO2, we're burning it like crazy. Up it goes, up, 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 up. What I think is really interesting is that this point here was done by an individual who I've met. Sadly, he recently passed away, Dave Keeling, a single scientist, a physical chemist, trained at Caltech, worked in California, the Keeling curve, this is called. And he made that first measurement around the same time CRISP published his paper. What most people don't realize is that most of the CO2 in the atmosphere, about a third of it's gone. We can't find it. Where did it go? It went into solution in the world's oceans. That's the blue line. And when you put CO2 into solutions, all freshman chemists know this, it becomes more acid, and hence ocean acidification. The pH changes. So what I want to say is the following. Anytime I talk about this, somebody says to me, well, isn't it over? What's Donald Trump going to say about this? Here's an interesting little article. California may follow Wales here and do a Cal exit. There's a lot of conversations. I don't think it's going to happen. But anyway, I said, hey, wait a minute. We're the sixth biggest economy in the world. Wah, wah, wah. We have all this money. We do this. We voted against him anyway. So there's a lot of buzz going on in the state of California about going independently. But whatever happens with the politics, what can a scientist do? Well, we study these things. And this is the last comment I wanted to make about this, is that what it costs to do all this has been speculated for years, and to my knowledge, this is the first study where actually we measured biochemically and thermodynamically how do you deal with the stress of ocean acidification at a molecular level, published just last year by my lab team in the Proceedings of the National Academy of Sciences. How do you maintain homeostasis under stress? What are the thermodynamic bases of being able to adapt to this? This matters a lot because it's the basis of probably producing food in the future. And I know there's some people in the audience here who have strong interest in the concepts of food from the ocean. So I'll just finish up with this. When CRISP was thinking about these things back in 1950, there was almost no aquaculture. Craig Kensler introduced me to aquaculture, and how to study it scientifically was introduced to me by Dennis Crisp and the NERC unit. But look what happened just recently. For the first time in human history, that's saying something, our ability to, quote, farm the ocean exceeds our ability to basically pillage the ocean through fisheries. There's a lot of parallels, and this is really to finish my thoughts towards students who might be up here one day giving one of these retrospective talks decades from now. The recent issue of Science on the front page had this challenge of how to feed 9 billion people. Students, by the time you're my age, there might be 9 billion people on the planet. That is a need for food that's 30 to 40% more than we have today. We don't think that can be done just from land. Many feel we'll have to think about using the oceans in different ways. In the same way that we think about seeds, corn, seeds. I remember at a lecture here in Bangor, professors of botany and professors of zoology talking about larvae as seeds. I remember Dennis being in that conversation, and I was a student thinking, damn, that's really interesting. If you could think of these little larvae as seeds that populate the ocean, could we make different kinds of seeds that could be put in different parts of the world, just like we do in agriculture? That could grow better in Bangor than in Wales, or in Scotland, or in this part of the world. It's very complicated science, and to finish, this is basically what I do. I won't go into any of the details. I will talk a little bit about it tomorrow, scientifically, about how organisms may be able to adapt to change at the School of Ocean Sciences seminar tomorrow. Basically, it's genome science. I have thousands of genes. They all go through something called the environment, and then they make something called phenotype. Genotype by environment equals phenotype. We're testing these ideas on continental scales. I'm part of a big program at the National Science Foundation where we work with industry, and I know this is a very important theme here in Bangor, to get academic and working with industry. All these red dots you see here, scanning the entire, from Mexico all the way up to Alaska, are aquaculture operations that we're working with, and we're saying maybe the seed, i.e. the larvae, that we've just made might work better here. Maybe they're going to be able to handle the situations in Mexico where the temperatures are warmer, a little bit different than Alaska, or something in the middle. And on top of that, ocean acidification is pouring in from the Pacific, hitting the coast. So the western United States have become one of the, quote, canary in the coal mines for dealing with some of the oceanography of ocean acidification. So Dennis. Amazing. I remember driving with him from San Francisco to Los Angeles after a conference, and he said, stop here, I want to look at something. I said, I don't think we can, because firstly, it's a marine reserve and it says no parking. He goes, oh, they won't mind, stop here. So, of course, I stopped. He goes down to look at stuff. Police pull up behind me and say, what are you doing? You can't park here and it's a marine reserve. And I said, well, I'm trying to help Professor Crisp. And so he came up and sweet-talked them out of the problem. The point I wanted to make, however, is that as we look at issues of biodiversity, we have to think about it in different ways. As I said, it's hard to break old habits, isn't it? It's very hard to break old habits. Professors will always say, my work matters, my way of thinking about things matters. But Crisp wasn't that way. I think he would have been shocked at this paper that came out to say that 91% of all species in the ocean have not even been described yet. That's an estimate. It's a statistical, mathematical estimate. That's a challenge. You won't find them maybe on a rocky shore. We have to think in different ways about diversity. At the same time, we have scientists saying the sixth extinction is here. Doom and gloom. Massive extinction. Wait. If you've only measured to 100, what about the other million or 10 million you haven't even looked at yet? How are we going to get our heads around these giant problems? We need to solve these problems, and as students, this is an incredible area to think to go into. You have front page of Time magazine saying climate change is causing new ages of extinctions, but on what? Maybe we're not even measuring the right things. A lot of people will give you advice on knowing stuff, on how to get there, what path to take. This is a road near where I grew up. It's a simple road in Ireland. You see many here too in Wales. The simple way is this. The simple way is that. I then went to Los Angeles and the roads look like this. They're not that simple. They're twisty and turny. This one is a little photoshopped up to make it even a little more complicated. But the concept is, I think, clear. Simple ideas are great, but flexibility in the fact that old ways may not work anymore. And many universities, to be blunt about it, are not equipped to handle these kinds of problems of the 21st century. They still have departmental structures that were set up basically centuries ago. And how we deal with real interdisciplinary thinking for things that really matter, students, this will be the challenge for you. Or as I like to say, just make your own road. If you don't like the road that your advisors and your professors give you, make your own. And I think a place like Bangor will help you make your own like few universities in the world can. Certainly it did for me. So when you ask students, it seems like there's a lot of challenges out there, a lot of negativity, problems with funding, grants, climate change, economic, politicians, this, that, and the other. What can one person do? Read about Dennis Crisp and others. One person can do a lot. Thank you so much for the opportunity to talk tonight. And I'm glad my wife and I are out of Los Angeles. Here's what the traffic looked like last night as they're all trying to go out to a Thanksgiving holiday. I'm glad I'm in Wales. Thank you so, so much for the honor to speak this evening. Thank you. Inspiration. You're very good. Yeah, if we're short on questions, we can go to dinner anytime you like. Yeah. Oh, yeah.
Thanks, Donald. That was a truly inspirational talk. There can be no doubt about that. He said we can take a few questions, so is there any questions in the audience? There's a couple of microphones that are roaming. Beth Ann at the front and Peter's got one there, so is there any quick questions? I will start the ball rolling in that I asked Tony Fogg, who you mentioned when I first came to Bangor, if I'm going to be successful, what is it I should study? And he said, rheology is what you need to study. I didn't understand what rheology is. So just for the students here, I mean, not explain what rheology is, but if somebody was to ask you the same kind of question, what would you say?
What I would say, especially to students, is to take a quantitative approach to the life sciences. People call it Q-bio, shorthand. A quantitative approach, bringing in the scale of thinking. It isn't just enough to count to 5 or 10 or do a mean and a variance. The scales are on millions. The data points are on billions. The number of species are possibly as high as 100 million. By some estimates, they could be 1 trillion. So you have to be quantitative to get your heads around these problems. So take as much math as you can, take as much physics as you can, take as much physical chemistry as you can, and then go into biology. Graham, you'd like that, right?
So there's a camp over in that side of the room that is smiling greatly with that answer, that's for sure. They're the physical oceanography team. Is there any other questions from the audience?
But I might say, the question is, can physical oceanographers talk to the life scientists? It has to be a two-way street. You would agree with this, right? That's what our Nature paper took us years to be able to speak the same languages between the hydrography of deep sea flow and the biological predictions of lifespans. Yes, but...
Okay. Yeah. Okay, I don't think there's going to be any questions coming from the audience. One of the most remarkable things of tonight, actually, was when you met an old colleague of yours, Andy Beaumont.
Yes.
I don't think you'd seen each other for how many years? Decades, at least. And it was an instant recognition. And then it was quite interesting to see where the discussion went immediately. Andy said, do you remember when we last saw each other in the lab, when we were totally out of our heads, or words to that effect? And I get the feeling that tonight, not that you're going to get out of your head, but I mean, I get there's going to be a lot of stories and there's going to be a lot of talking about the old times tonight, but we're very grateful for what you said to us, for coming all this way, especially on Thanksgiving night as well. So thanks very much, Donald.
My pleasure. Thank you very, very much. Thank you. Thank you. Thank you. Thank you.
Great. Thanks. You take the microphone off now.
Oh, yeah.